My dear students, today talk on vectors, types of vectors. Genome size varies among different organisms and the cloning vector must be selected accordingly. For a large genome, a vector with a large capacity should be chosen so that a relatively small number of clones are sufficient for coverage of the entire genome. Genome nothing but a total sum, a sum total of genes found in the individual. However, it is often more difficult to characterize an insect containing in a higher capacity vector. Vector means it is one of the carrier. It carries foreign DNA fragment. It helps in the recombinant DNA technology. So, few examples for vectors. Below is a table of several kinds of vectors commonly used for genomic libraries and the insert size that each generally holds. So, type of vectors and then insert size. So, first one now plasmid up to 10, thousands of bases here now. Plasmids are particularly up to 10, phase lambda up to 25, cosmids up to 45, bacteriophage P1 70 to 100, P1 artificial chromosome 130 to 150, bacterial artificial chromosome 120 to 300, yeast artificial chromosome 250 to 2000. So, nearly about thousands of bases carrying from one cell into another cell. This is something about definition about vectors and types of vectors, particularly plasmids, phase lambda, cosmids, bacteriophage, P1 artificial chromosomes, bacterial artificial chromosomes, and yeast artificial chromosomes. I'll take first one now, plasmids. A plasmid is a double-stranded circular DNA molecule commonly used for molecular cloning. So, plasmid can be defined as it is a double-stranded circular DNA molecule commonly used for molecular cloning. Plasmids are generally 2 to 4 kilobase pairs in length and are capable of carrying inserts up to 15 kilobases. Plasmids contain an origin of replication, allowing them to replicate inside a bacterium independently of the host chromosome. Plasmids commonly carry a gene for antibiotic resistance that allows for the selection of bacterial cells containing the plasmid. Many plasmids also carry a reporter gene that allows researchers to distinguish clones containing an insert from those that do not. Second one is phase lambda. Phase lambda is a double-stranded DNA virus that infects Escherichia coli, E. coli. The phase lambda chromosome is 48 0.5 kilobase space long and can carry inserts up to 25 kilobase space. These inserts replace non essential viral sequences in the phase lambda chromosome, while the genes required for formation of viral particles and infection remain intact. The insert DNA is replicated with the viral DNA, thus, together they are packaged into the viral particles. These particles are very efficient at infection and multiplication, leading to a higher production of the recombinant lambda chromosomes. However, due to the smaller insert size, libraries made up of lambda phase may require many clones for full genome coverage. Cosmids. Cosmid vectors are plasmids that contain a small region of bacteriophage lambda DNA called the COS sequences. This sequence allows the cosmid to be packaged into the bacteriophage lambda particles. These particles containing a linearized cosmid are introduced into the host cell by transduction. Once inside the host, the cosmids circularize with the aid of the host DNA ligase and then function as plasmids. Cosmids are capable of carrying inserts up to 40 kilobase pairs in size. Next one is bacteriophage P1 vectors. Bacteriophage P1 vectors can hold inserts 70 to 100 kilobase pairs in size. They begin as linear DNA molecules packaged into bacteriophage P1 particles. These particles are injected into an E. coli strain expressing Cray recombinase. The linear pi vector becomes circularized by recombination between two lax p sites in the vector. P1 vectors generally contain a gene for antibiotic resistance and a positive selection marker to distinguish clones containing an insert from those that do not. P1 vectors also contain a P1 plasmid replicon, which ensures only one copy of the vector is present in a cell. However, there is a second P1 replicon called the P1 lytic replicon that is controlled by an inducible promoter. This promoter allows the amplification of more than one copy of the vector per cell prior to DNA extraction. The diagram represents the nature of bacteriophage P1 vectors. So you're going to observe the region of origin. In that region, we are going to insert the foreign DNA segment for the purpose of cloning. Next one is P1 artificial chromosomes. P1 artificial chromosomes have VHs of both P1 vectors and bacterial artificial chromosomes. Similar to P1 vectors, they contain a plasmid and a lytic replicon as next one is a yeast artificial chromosome. Yeast artificial chromosome are linear DNA molecules containing the necessary features of an authentic yeast chromosome, including telomeres, a centromere, and an origin of replication. Large inserts of DNA can be ligated into the middle of the egg so that there is an arm of the egg on either side of the insect. The recombinant egg is introduced into yeast by transformation. Selectable markers present in the yeast artificial chromosome allow for the identification of successful transformants. Yeast artificial chromosomes can hold inserts up to 2000 kilobase pairs, but most yeast artificial chromosome libraries contain inserts 250 to 400 kilobase pairs in size. Theoretically, there is no upper limit on the size of insert a yeast artificial chromosome can hold. It is the quality in the preparation of DNA used for inserts that determines the size limit. The most challenging aspects of using yeast artificial chromosome is the fact they are prone to rearrangement. Any clarification, any doubts about vectors, types of vectors, send me the messages. Thank you.